Thank you very much. Hello. Good morning, everyone. Thanks for staying for the last day of the meeting, and big thanks to the organizers for setting up such a great lineup. It's really, the whole meeting has been a pleasure for me, as most of the talks were quite relevant to what we do in the lab. What I will talk about to you today is a story that started in the lab maybe 3 or 4 years ago when we started looking at high C data. But in this talk I'll kind of start to maybe position myself in the context of these problems, maybe kind of coming back to Tuesday's discussion to kind of give you the idea of whether I'm more into fun of analyzing high C data or more into kind of trying to be predictive. So I think I will not need much of introduction of the data itself, but I'll give you a first few slides, the idea of where I come from, and then I'll discuss two methods that we have developed for this task, and give you an outlook for applications. This is all unpublished, but the code is finally out, and if somebody wants to use it, it's actually available. Okay, so where do I come from? The focus of my lab is basically gene regulation, and I started very theoretically as looking at models of gene regulatory networks, and it was quite interesting to look at this in the context of development. But naturally, as I tried to look more and more closely at the data collected from animal tissues, you figure out that this is more complex than just a transcription factor like MEF2 going after, let's say, its target of beta tubulin. There is much more mechanism where we need to understand to actually have some kind of semantic for this kind of diagram. There we go. So naturally, the enhancers came in, and I spent quite some time in Eileen Furlong's lab looking at enhancers. In this case, it is in Drosophila, and kind of trying to understand how a given gene is regulated by a number of regulatory elements that are scattered somewhere on the chromosome, usually on the same chromosome in the vicinity. And this kind of made the task of understanding gene regulation a little bit more modular. These are discrete elements to a large degree, and as far as we agree on some sort of definition of that's probably a compromise from the reality, but we can actually understand the semantic of regulation of a certain transcription factor better when we know that this regulation is mediated through a number of discrete elements that are driving different parts of the activity of the gene. But to actually still see whether we understand how this works, we need to make models that actually put these things together in the context of chromatin. And to this extent, I was involved also in looking at models that try to actually go and predict gene expression. In this case, it's not so much a goal of actually having a tool to predict gene expression, but maybe, for me, this was more about trying to understand which parts of this image. So in this case, we have genes with their location on the chromosome. We have some proteins binding to the chromosomes. In this particular case, these were insulator proteins, and transcription factors. And then those transcription factors end up being in some modules. And then some of them have some known activity, and then you have also histone modifications that are telling you something about the global activity of this situation. But it's not so much. We built a model that was probabilistic and was kind of was integrating all of that to give a predictive method for trying to see where a gene is going to be expressed. But it was much more of a exercise in trying to see whether you gain or lose something by adding any of these particular pieces of data, and see how important they are for the accuracy of the prediction. So one of the things that at this time in 2012 was maybe not so common to do was to look at the insulator proteins, and we found that they were really instructive in case of Drosophila genome to see So you have, for example, this is a locus where two important regulators sit. So Tinman and Bagpipe, they're both involved in mesoderm differentiation, but they do different things. And then when you want to understand all of these blue rectangles are actually putative regulatory elements. If you want to understand which of those are talking to which of the target genes, it is actually quite instructive in this case to look at the chip. This was still chip data, I think. In this case, I think this is CDCF, but there's a number of other proteins that are occupying this spot, and this spot is a boundary element. So if you would remove it, then the regulation of this region would become more complicated. So we have put this into our mathematical model, and it helped us make predictions about where exactly this gene and the other one will be activated. So the important thing that was already mentioned here several times is that these boundary elements. I mean, one of the things is they somehow end up recruiting a number of different proteins which help them mediate these functions. We've heard from Leonid about the loop extrusion and there's a lot of discussion in the field about how exactly this is working. We don't have exact ways to look at this, but one of the things that is clear that they are not only separate things from one another, but they also, so this is one example from Drosophila, where it is well known, from, I think, from the 90s that this sex-coms reduced gene is regulated by elements that are beyond the loop that loops out FATs with its regulatory elements. So these boundary elements, they mediate contacts as well as separate regulatory environments. And with my student Pavel, we've looked at those, and we were wondering whether we can also, similarly to some approaches in predicting regulatory elements, whether we can actually also predict these boundary elements. And long story short, using supervised learning, we have been able to find the locations of those boundary elements with a relatively good depending. So from the sequence alone, you can get about 85% of the area under the curve. If you add histone modifications and proteins, you can get more than 90% accuracy. So this is pretty good. But we still don't understand how they're connected to each other. And for this, you need really high-C data. And I hardly have to. Yes?
I'm just curious. What does it mean to predict an insulator? So what is the what are
location of where the So in this case, our training set was actually from a high-C data, from Sexton et al. Paper, where they actually looked at the basically topological features of the chromatin. So this is actually exactly these lines here. So they took high-C data and delineated where the boundaries are. They had about 1,000 of those in the genome, where they were confident that this is really a boundary in topological sense. And we were wondering whether you can predict this, for example, from sequence. And in 85% of the cases, you can actually predict them from sequence. So that this is going to be a topological boundary.
Just sequence, no.
Just sequence. I mean, if you add chromatin marks and proteins, then it goes up to 90%. So this is not really the main topic, but but we've been looking at this as well. So it has been shown also by other people, and just to kind of remind you from what Leonid was and some other people were talking about before, is that there is these two ways of looking at these high-C data. So I'll not go into details of what exactly these matrices are, but already in Lieberman-Eiden paper from 2009, it was kind of observed that there are these A-B compartments. So if you look at the principal components of the signal of the contact matrix in the high-C, you get these kind of clear checkerboard patterns, and these correspond to large extent with the activity of the genome. So you have this more active and more inactive compartment, and bless you. And the other approach, which is kind of at the same time the way to look at this, is to look at those heavy rectangles in the contact matrix, basically looking for the domains, and there can be some hierarchy seen in this. So basically, you can have a very dense region here, and this is a part of a larger dense region, and this whole thing is actually a dense region taken out from a part of the chromosome 3R. So this is no news, I guess, to everybody here. What is maybe less advertised in the papers, but it should be quite natural, is that this is really two ways of looking at the same data, and the way we see this in papers, so kind of to rephrase what Remo is saying, I mean, with heat maps you can pretty much show anything from your data. So in this case, this is the very same data, but depending on whether you actually look at this in just the contact frequency or whether you make a covariance, a normalized covariance matrix from this, you're getting a very different picture. And it also, because we don't exactly know what we're looking for, and we don't exactly know, we don't have a gold standard of what exactly does it mean to segment chromosomes, we are kind of, I mean, when we read paper, it definitely applies to me. When I read papers, it kind of, my brain gets into, when I see maps presented this way, I kind of look at the very small contacts. When I see maps presented this way, I kind of, it's more driving my thoughts to the compartments. What I wanted to emphasize is that actually both these things happen, and as Leonid was saying, this is usually more large-scale objects, and these are usually smaller scale. But in fact, there is some overlap. So there is some, for example, this is a larger compartment, and within it there's still AB kind of, you still see the checkerboard pattern, and similarly here you have a denser area with a checkerboard pattern in it. So it's a hierarchy, and it has been postulated as early as in 2009 that this clearly is a hierarchical structure that we're looking at. So there has been a number of tools. I'm not really trying to be comprehensive here, but it's definitely a large number of approaches that has been available. In particular, so I would basically divide them into two groups. One of them would be including the scaling factor, the directionality index, and the Armatus tool from Carl Kingsford group. Similarly to the statistical potential, one of them is really looking at those heavy, so to speak, heavy elements close to the diagonal. So the idea here is usually that, I mean, and this is a figure from Levy-Leduc paper from 2014. You usually have a certain model where basically you have rectangles that have a different contact frequency that are close to the diagonal, and then you have a less frequent contacts outside of this. And the idea of segmentation, the cost function is to find the segmentation that is basically has the most heavy rectangles at the diagonal and the least heavy other part. It's the directionality index and scaling factor are slightly different, but they actually look at the same thing, just a little bit differently. So the people, some people might recognize this is actually looking at the left to right ratio of contacts, but it indeed is looking for those triangles. So kind of one more word about Armatus. So this is, to my knowledge, the first approach that was openly trying to at least pose the problem of finding the hierarchy of those domains. It is actually using a parameter. So there is a gamma parameter that you can tune. And if you tune it, then you basically get different segmentations. You can see they're mostly either agreeing or there is aggregation of domains. It's a little bit tricky because there's no clear idea of how does the gamma parameter map to the resulting size of the domains. But it is, I think it definitely deserves credit for the first to state the problem. Recently, more recently, there was a paper from Anna Pombo and Mari Nicodemi lab where people have been looking at the differentiating cells, and there the approach was actually to use TAD segmentation, flat segmentation as a first approximation, and then aggregate them up into so basically you look first for the heavy triangles, and then you look for heavy rectangles between them, and then you get a tree-like structure of the domain. So it's an interesting case where this is basically using the hierarchy above TADS. So this is, instead of using this kind of PCA-like algorithms that actually look for checkerboard pattern, this is actually looking for super TADS or meta TADS, however you like to call them. So what we started to develop, actually, the master student who was responsible for that is Krzysiek, who is sitting here because he now works at Google, and it was a short drive for him to actually listen what's going on. So this is what we now call Sherpa, which stands for Simple Hierarchical Profile Aggregation. And I won't really give you details, but kind of the basic idea is you start with the matrix, you go into covariance or normalized covariance matrix, where basically you can think of every entry in the matrix as a Pearson correlation between two vectors. So kind of this boosts the signal, and it kind of we are using information about the context from the whole vector instead of the just this one Mij entry. And then you can just use hierarchical aggregation. You basically look for the two nearest vectors, and then you aggregate them, and so on and so forth. And the question is, where should you stop doing this? And to find where you should stop this, we actually devised a relatively simple trick. So basically, as you look at how the Pearson correlation coefficient changes as you move up the tree, we found, kind of through observation, that it usually shows first a certain number of high correlation merges, and then it drops down. And the kind of null model is here that actually this should be actually a flat, a linear decay. So what we're doing is we're basically trying to see, first of all, whether we see this kind of enrichment, so whether there is actually a population of merges that is actually better than the kind of normal flat decay. And if we see that the bilinear model is fitting better than the linear model, we are actually choosing this inflection point, and we're cutting here. So basically, we aggregate everything here and do not aggregate those. And this is the one step of Sherpa algorithm. And then we merge those vectors and average them, and then we basically you start over. So you calculate Pearson correlations of those average vectors, aggregate them, find the inflection point, and so on and so forth. And this is what you get. So kind of the way this is a fragment of a human chromosome, and what you get here, these blue triangles is the output of this algorithm. The green here is an output, I think, of Armatus. And it really does agree with majority of those tools. What we arguably get is we have a lot of more in the lower spectrum. So these kind of high resolution things that we get, mostly due to the fact that using this kind of normalized covariance matrix instead of just searching for heavy triangles. Yeah.
You're doing Pearson correlation on this plot on the left here. I don't understand what the correlation is being computed. Are you aggregating within a subtree? Not the top one, the bottom one, where you're changing the threshold. So at a particular threshold, what does it mean to compute a Pearson correlation?
No. So it's at every merge. So let's say the most correlated are vector 1 and 2. And they have a correlation of 0.95. Okay? So if I was cut this tree here, this would be just a very beginning, and it would be at the Pearson correlation of 0.95. And here maybe this is a correlation of 0.7. Okay? And this means I have merged 150 pairs of vectors, or pairs of clusters.
So the correlation between the subtree with 1, 2 and the subtree with 3, 4, 5 is sort of the average of the... at the C boundary that you drew in the top right.
Right. Yep.
Yeah. How do you compute the correlation at that level?
So this is the last merge that we made. So this is basically, and we're using complete linkage clustering. So basically, we're guaranteed that if we cut at correlation of 0.75, that we don't know what level we will be doing. We can, yeah.
Averaging the correlations. How are you computing that number between those two things?
Yeah, so this is the complete linkage. So basically, this is the lowest correlation between, so if you have two clusters that you're merging, this is basically the lowest correlation between any two vectors in the clusters.
And then another follow-up question. So you're saying that you get many more fine-scale resolution, right? But aren't other hierarchical methods, don't they have a parameter that they could tune to do that as well? Or is
it's not so easy. It's not so straightforward. In principle, yes, but it's most of the methods that use the kind of heavy triangle kind of approach. This is not so straightforward. So, for example, directionality index or Armatos, it's not so easy. I mean, I'm not saying it's not possible to do. It's definitely, upon certain, you could definitely make them be very selective. I think without going into this kind of correlation matrix instead of the actual count matrix, it's a bit tricky. So kind of a little bit bigger, you can actually do this on the scale of whole chromosomes. It gives you some large-scale elements, some small. It's kind of, this is how it looks like. Yeah, 5 minutes. Okay, so that's going to be quick now. So this is the Sherpa algorithm, basically how it works. The other approach is kind of what we found is that these large hierarchically aggregated things, once they hit the level of these AB compartments, they're not really very useful. So what we started developing some time ago is the other kind of complementary approach, which we called OPPA, which is kind of optimal PCA-like aggregation. So this is a dynamic algorithm. This goes top-down now. So since you remember these checkerboard patterns, so what we want to optimize now is a segmentation that not only has heavy triangles along the diagonal, but it also has a checkerboard pattern. So actually, I want to see negative signals. So I want to actually, this is a penalty. If I have heavy elements in this place, this actually is a penalty for me. So if I decide to put a boundary here, I will actually penalize for all that. And then if I put the next boundary here, then I'll penalize for this, and I want to have more contacts in this area, right? So this is specifically going for the checkerboard pattern, and it goes completely opposite. So kind of it's top down. It's a cubic in terms of the size of the matrix. So it's the length of the chromosome. So it's not the fastest, but it's polynomial, and you can actually find the optimal solution, assuming that it doesn't go well if you have breaks. So if it's not happening in the data, but if you had actually, in theory, you could cook up a matrix that would not work, that this algorithm would not work on it. But if you assume that this is somehow noisy, proper compartment signal, you actually get those. And the nice part about this is that when you run both of the tools on the same matrix, what you get typically is Sherpa is giving you the small stuff, and OPPA is giving you the larger part. So kind of maybe to underline this, both of these methods will stop naturally at a certain point. So Sherpa will stop when there is no longer an improvement in aggregating. So it usually stops at these kind of PCA compartments, because now the next neighbor from you has really the opposite vector, right? So it might actually still have the contacts, because if you use those kind of heavy diagonal stuff, then there will be a lot of contacts between this compartment and the next compartment, because they're just on the neighboring on the chromosome. But the actual correlation of those vectors will be negative. So Sherpa will stop at this point usually, and OPPA will also stop at this point, because then there is no longer the checkerboard pattern, is not so prominent inside. But you also see some places that they actually overlap. So for example, here Sherpa can aggregate this into a larger domain. The same thing you get from OPPA. And kind of you can see that they overlap. And this is those places where you see clearly domains aggregating with the checkerboard pattern inside them. So this is something that we have been struggling quite a bit. We didn't quite solve it. These kind of cross-like patterns didn't quite solve it with Sherpa. But it seems we can do this. So very, very quickly about applications. None of this is published, so I'll not really give you depth, but kind of just to advertise what you can do with this. One thing is you can compare two different, so maybe this is not so obvious, but these are two different contact maps, and you can see that when you run Sherpa on them, you get different results, and this really reflects, for example, here you can see this domain really does look like it has the same structure in both experiments, and here, for example, you see clearly that these two domains are aggregating together, while in this experiment they're not aggregating together. So you can see these kind of rearrangements. They mostly happen at a very low level. So it's not really those this is below TAD level. So kind of to give you, if we run directionality index, we would get kind of this thing as a domain, and it wouldn't really let you look at those kind of rearrangements, which are, I mean, it's not exactly high resolution data here, but this is kind of giving us hope to look at kind of switching from one enhancer environment to another enhancer environment by a gene. This kind of, this is where it leads to for us. The other thing that we've been working on, and we have some promising results, hopefully to be published soon, is looking also at long-range contacts. So now that you have all these domains, this is a very nice setup for hypothesis testing about kind of having pairs of domains that are in contact, specifically in certain cell type or not specifically in certain cell type. So it's kind of it's been a workhorse, and we've studied a lot in terms of finding these kind of regions that are not. I mean, this particular one is kind of visible. So this is the clean signal you get from statistical testing. This is the kind of the actual contact matrix. You can see this, and if you know where to look, you will actually find that this blob is not appearing in the other contact matrix.
How do you distinguish that from structural variation?
Structural. That we don't, yeah. I mean, one structure, if you really think about, so I wouldn't go with this into cancer cell lines where I would expect many chromosomal reorganizations. I don't know what I would do. I think that this is I don't think anybody has solved this kind of thing yet, but I mean, we definitely didn't. But if you have a differentiating cell line, I mean, in principle, you don't expect these things to be reorganizing their chromosomes as they go. So I think it's, I mean, we're getting some meaningful enrichments in terms of chromatin state at those places.
That's actually a great application for detecting these kinds of things. It's actually detecting structure.
Yeah, it's unclear whether, how much this is in agreement with the domain structure, because here we're only testing hypotheses about certain domains at different levels being in touch. But it's, yeah, so we've also looked at gene expression distribution over chromosomes, kind of quickly. It does give you an edge over TADs because you can really look at the lower level, and it is agreeing, even though we have certain data sets where there's very different genes that are differentially expressed. All of them, the variance is very nicely distributed along our domain structure. The other thing that we've also looked at is the co-expression across species, and again, this is reliably at the lower end of the spectrum. There is more of a difference between random and random control and what we get. So kind of to summarize, what I'm trying to sell you here is a set of tools and set of approaches. It's by no means this is the only way to do this, but I think that we have, after a few years of work, we have found a way to kind of marry the kind of top-down and the bottom-up approaches of the PCA and the kind of looking for those heavy triangles. And in particular, in this case, if you want to just remember one thing that is useful is it's, I think, using these correlation matrices is really a useful thing to do. Yeah, so this is. Yeah, there might be downstream implication. One more plug is that this is this year in autumn there will be a school on computational approaches to chromatin organization. So if you are interested in these things, there will be a lineup of I think excellent speakers. There will be fellowships, so we're trying. I know there are some people who have been in the very same place 3 years ago when we did transcriptional regulation school. It will be the very similar setup. We would try to get fellowships for all the students. If you want to come, just let me know. There is a link here. And finally, credit. So the initial implementation of Sherpa was done by Krzysiek, who is actually sitting here. And Paweł has been mostly. So this is Paweł. Paweł was mostly involved in insulator prediction. Rafał and Irina are now, so Rafał is our go-to guy for high-C normalization, and he's done a lot of these experiments with gene expression correlations. Irina is doing mostly from the stuff I talked about is is doing the long-range contacts, and Ilona was involved in another gene expression study, and we are collaborating with a number of people. In particular, Eileen was giving us data for Drosophila, High-C, and Mina Kaikkonen from Helsinki, from Kuopio, sorry, was kind enough to share with us High-C data for HUVEC cells. And then with this, I might maybe take 1 or 2 questions. Thank you very much.
So in this higher resolution partition, do you get any kind of validation that this is biologically meaningful? For example, in the beginning you had this idea of looking at insulators. So do you get more insulators or better specificity at the higher resolution?
So one thing to kind of to be clear about here. So when I'm talking about higher resolution, so we haven't really gotten yet to these, because there is this RAW and other data set that is really deeply sequenced human High-C. When I'm talking about high resolution, so for example, what we use in the human data set, we're using at bin sizes, so a single pixel in those images was on the range of 50 to 150 kb, right? So this is not really high resolution in terms of enhancer promoter, single enhancer promoter interaction. So we're not really replacing capture High-C or Chiapet or whatever you might want to really probe those so singular. But in terms of validation of this lower level, this is really kind of, I just showed you this one plot here, but this is kind of, so this is a plot of how the observed correlation of expression goes as opposed to randomized control. And it actually is peaking at the very lower end, and we have control, so this is not due to the fact that these are just smaller elements and these things are closer on the genome. So we have indications. I wouldn't say that this is completely proven, but we have, for example, also looked at replicate reproducibility, and it seems to be not getting worse at the lower level. So these kind of things make us that this is really, we're looking at something that is real and not just kind of dividing arbitrarily those segments that other people have.
Okay, maybe one last question.
So if I understand right, Sherpa is bottom up and finds small scale, and OPA is top down and finds large scale. But both of them, you could remove your stopping criteria, right? And they could find complete all scale.
No, but they have in the end different criteria.
That's my question: is when you get rid of the threshold and you have them both remove everything. It sounds like what you're saying is that you believe that the ones that OPA finds at small scale are better than the ones that Sherpa, other way around.
Yeah.
Are better in some sense, and the large-scale ones are coming from OPA.
It's not so straightforward to remove these stopping criteria. So kind of, in particular, I don't have anything to point to now, but...
Pearson correlation threshold, and you just say continue to split, and keep splitting until you get down to one pixel.
No, but in order to split, the top-down in particular, it has a possibility to just tell you this is a one triangle. There is no checkerboard pattern in it, right? And it won't go further. There is no reason, because the cost function is this checkerboard pattern. So in order to have a split, you actually have to have a negative signal in the above the diagonal. And this actually leads to the situation where we're not. It just stops. It just cannot go further. And it happens to be about the same region where the Sherpa is. We could go, but it's still, we're trying to find this inflection point. And if this is linear, there's really, this is just kind of turning into random division, because then you have a noisy linear signal and you're trying to just find some blip. So indeed, removing the stopping criteria would mean that you kind of, you're just randomly splitting. So we don't do this.
Okay, let's thank Bartek again.